Good afternoon, everyone, and a very warm welcome to you all. I have never had an audience that has been so polite and gone so quiet so nicely, so thank you. For those of you who don't know me, I'm Selina Wray, and I'm one of the Deputy Directors of the Institute of Neurology. And it's really my pleasure to be the host for this evening's inaugural lectures. It's really wonderful to see so many colleagues here, but more importantly, I'd like to extend a very warm welcome to friends and family of our two new professors who are here joining us to celebrate today. Inaugural lectures are really a special occasion. It's where we come together to celebrate an individual's academic and professional achievements. But more importantly, and what's really nice, we get to find out a little bit more about the person behind the research. We get to hear a little bit about their journey, about their stories, about their successes and their challenges, the journey that brought them to this point, but also it gives us an opportunity to reflect on how that's influenced them and the way they shape their field and also our institute. We're really fortunate today that we will hear from two colleagues who are both leaders in their respective fields, and I'm really excited to hear more about what's brought them to this point in their academic journey. So I will keep my introductions very brief so we have as much opportunity to hear from them as possible. So our first speaker today is Professor Hani Marcus. Hani is a consultant neurosurgeon at the National Hospital for Neurology and Neurosurgery, and he specialises in minimally invasive and endoscopic surgery, I hope I'm saying all of these words right, for pituitary and anterior skull-based tumours. He's done academic surgical training in Cambridge, Imperial and UCL, and also undertaken fellowships in Calgary and Zurich. And he's established a research programme which is focused on artificial intelligence and robotics in neurosurgery. Hani has been the Deputy Director for the Wellcome EPSRC Centre for Interventional and Surgical Science, which develops new surgical technologies, and he's currently the Interim Chair of the IDEAL Collaboration, which develops frameworks for their evaluation. If that wasn't keeping him busy enough, he's also the Chief Medical Officer for Panda Surgical, which is an artificial intelligence and surgical robotics spin-out. And he also shares his knowledge with the next generation of scientists through his role as the Director of the UCL MRes Neurosurgery Programme. Please all join me in welcoming Professor Marcus to deliver his inaugural lecture.
Thank you very much. I'm getting strong wedding vibes seeing all my friends and family here. Feels like I'm about to give a speech. I'm just missing the champagne. So the title of my talk today is Seeing and Doing. And as some of you may have figured out, that's largely because throughout my journey, I've been seeing mentors and trying to do what they do. And you'll hear about some of those mentors shortly. But it's also the nature of my practice as a surgeon. Fundamentally, all that we do as surgeons is see tissue, decide what we need to do next, and then do it as best we can. And my research, which as you've heard is around AI and robotics, is using AI to understand what we see better during surgery, to make better decisions, and using robotics to execute those decisions more faithfully. Now, perhaps the most obvious way to tell this story is chronologically, but there is another way to think about it, which is around the three interests that I've had for a very long time that over the years have become better defined and more recently have started to converge. Those are, of course, surgery, technology, and evaluation. And the first of those began with my dear mother, who's in the audience just on the far left there, and is pictured here on the left side of the screen with my sister Diana and myself there in the image. Now, I had the good fortune of both my parents being medics, and my mum in particular had a near obsession with her patients. So most evenings at home over dinner, my mum and dad would be talking about patients they saw. Quite often when my mum was working in hospital, she would bring us with her. And when my mum later trained as a GP, we'd quite often, my sister and I, be in that very car there, waiting for her while she visited her patients. And in a very unorthodox interpretation of the doctor-patient relationship, my mum would often invite patients she felt needed it to tea in our house. And decades later, I still remember her patients that she felt it needed cheering up. So I learned a great deal from my man, in particular, this idea that you really care for your patients and do your best for them. The next big influence of my early childhood was Khalid, my cousin, pictured there on the left with me as a young chap holding him. He's in the audience too. And Khalid was an older brother in everything but name and lived with us while he was here in UCL, in fact, doing chemistry. And I saw Khalid do that degree in chemistry and then do a PhD. And if you have a car now, the technology in your air conditioner is likely technology that Khalid built. He built it here in UCL. It became a company. He span it out and then has used that to really impact society. And so Khalid spent a long time talking to me about this idea that you could build stuff and have it affect people. This might not surprise many of you, but I was a geeky kid, as I am a geeky adult. And I, having been taught by Khalid how to code and build things, spent absurd hours doing this stuff at home, particularly trying to code for this stupid Lego Mindstorms that you see center. And I then inflicted this disease onto my younger brother, who you can see pictured there on the right. So he's the fan of personal displays of affection you see cringing as I'm holding him. And together we spent many hours coding. And the last big influence was from my father. You can see pictured on the left, the chap in the blue shirt, and to his left is Bob Edwards, Sir Bob Edwards. So my dad is a doctor specialising in fertility. He came to the UK from Egypt to help Bob Edwards in the first early experience of in vitro fertilisation. And to those of you in the audience who are young, this may seem surprising, but actually back in the 80s, when this picture was taken and I was a kid, in vitro fertilisation was really deeply contentious. So my dad was refused communion several times in church because the priest felt it was deeply unethical for him to be doing IVF research. So it really imprinted on me. And beyond the ethics, there were real clinical concerns. Again, some of you may know, but it's not commonly known, that the very first successful conception with in vitro fertilisation almost killed a mother. It resulted in an ectopic pregnancy. So the very first time it sort of worked almost resulted in a mortality. So there were really major concerns from a safety and effectiveness point of view, ethics aside, that we should be doing IVF. Now, I have to confess, sorry, Dad, that I really don't remember much of my dad during my childhood. He worked a lot. But those memories I do have often revolve around him coming home from the weekend with stacks of handwritten notes, observations from patients. And the resolution to this uncertainty over whether IVF is safe and effective was to do statistics. And so he would read off his handwritten notes, numbers, and I would punch them into the computer. And then at the end, because my dad is a man who believes in accuracy, we'd swap roles and I would read the handwritten notes and he would check on the computer if they were correct. And I never got co-authorship credit, but I still learned a lot. Now, at the end of school, I had to make a decision about where to go. And after a bit of indecision, I thought the best balance of these various interests was to go to a traditional medical school, Cambridge. And to those of you who know me, you may know this story, and if you don't know the story, this may not surprise you. But despite growing up mostly in Cambridge, I got lost on the way to the interview and turned up a few minutes late, so late, in fact, that the interviewer had to come down to find me on the floor of the building. And at the end of this interview, flustered as I was, being a young 17-year-old who'd never interviewed for anything in my life, I walked straight through the exit and then realised I'd walked into a walk-in cupboard. And I stood there for a few moments, deciding what my best plan of attack was, only to hear the interviewer outside laughing and saying, "You should try again," which I did. And despite or because of that, they offered me a place. And in Cambridge, I really became interested in neuroscience, both neurology, psychology, and neurosurgery. And the picture on the far right is myself with my mentor at that stage, Ramez Krolos. And this is a picture taken just 1 hour ago. He worked in Singapore and he came all the way here. I saw him just before this lecture. And yet he's not in the lecture theatre because as an Egyptian, he's watching the Egypt versus Argentina game. But from Ramez, I really developed this obsession with neurosurgery, particularly around the base of the skull. The anatomy was incredibly beautiful and complex, the surgery really intricate, and of course, most importantly, as you heard earlier, the patients benefit so immediately from neurosurgery. Patients will wake up in recovery and sometimes report that the disability has improved, that they can see or they can move where they couldn't before. So I was dead set on it. I was still a geek, still did lots of coding and playing with robots, and actually I found it incredibly helpful learning about neuroscience both clinically and from a basic science point of view to inform developing and building these coding networks and robotics. The main ideas I drew from biological inspiration for deep layers, multiple layers in a network, and having reinforcement learning, which are very much accepted now if you're in the field, were very contentious back then. But seeing it work in the brain, I felt convinced it was the right way to go. And I also, in my early foundation years, had lots of exposure to clinical trials in neurosurgery. So pictured on the left is Tom Santaris. He was a registrar when I was a foundation doctor in Cambridge. Took me under his wing, and there aren't many good examples of randomized control studies in neurosurgery, but he was doing one in Cambridge while I was there. And the most common operation you do as a trainee neurosurgeon is very simple in many ways. It's a hole in the head to drain blood. But even for a simple operation, there are so many steps. And the last step of this operation is whether or not you leave a drain. And the vast majority of neurosurgeons wouldn't, because it's an extra step, there's risk attached to it. But we did a study, and the result, to our great surprise, was in fact that if you did not leave a drain, the mortality rate was double. Now, this was published almost 20 years ago now, and the impact from this study has far greater reach than the patients I've treated myself, because practice has now convincingly changed. Almost all surgeons, if the brain doesn't reinflate, will now leave a drain. And the lives saved from this work that Tom led has really impacted patients globally. And so seeing Tom do the study and being involved with it myself really firmed up this idea that evaluation is very important in neurosurgery too. So I then took a job as an academic clinical fellow in neurosurgery in London at Charing Cross Hospital, part of Imperial. And that's where really the problem started to take shape. Now, I had the good fortune, or not, depending on your point of view, of the person who hired me at Charing Cross shortly after I was awarded the post, stopping working there. So I started at Charing Cross with no one really having a good idea of who I was or what I was supposed to be doing. And I had the gift of a lot of free time on my hands. And so I did what most people at that stage would do, which is I just thought of places I wanted to visit and then planned work trips around them. And the first of those places was to go to Zurich. So on the left, you can see pictured with Robert Reich on the far left of that image, and a younger looking version of me. And Robert Reich with Axel Panetsky literally wrote the book on keyhole neurosurgery. He wrote the book describing these keyhole approaches. And I have to say that when I saw Robert speak in Cambridge, lots of consultants who I really respected were quite dismissive of these endoscopic approaches 20 years ago. They felt it was making your life much harder for a surgeon, probably no safer, and maybe much less safe than doing traditional surgery. So I went to go see it for myself, and I was blown away. There were many patients who I saw who may not have been offered surgery in the unit I was working in Cambridge, but had surgery through tiny incisions, a centimetre or so in diameter, and would recover so rapidly that they almost looked like surgery hadn't happened. So I was very convinced that this was much better for the patient. But I was also convinced that it was an absurdly hard operation for the average surgeon to do in most cases. Robert was very uniquely experienced in doing these approaches. During his Bachelor of Science degree, he did hundreds of microsurgical dissections on cadavers. Later, as a resident, Axel Panetsky, who was developing these techniques, got Robert to be in the lab actually describing the steps, and he did another 1,000 cadavers. So by the time he became a consultant, his experience dwarfed mine as a 10-year consultant. So he was a very experienced surgeon, even at an early stage. Most of us can't get there. I then completely separately went on a trip to see Garnet Sutherland, pictured in the centre, in Calgary. And he at the time had developed the most advanced robot in the world, definitely for healthcare, maybe in general. And this robot is an incredible feat of engineering. So it operates inside an MRI scan. It's hard to build things that are MRI compatible, and has 50 micrometer precision. So that's about the size of a cell. So incredible feat of engineering, but it's completely ill-suited to minimally invasive endoscopic surgery. You'll see the instruments it holds in its hands are the same instruments that we hold as surgeons doing open microsurgery. So really impressive, but not applicable to this sort of operation. And finally, I continued to work with trials, and Professor Peter Hutchinson, another early mentor from Cambridge, stayed in touch. We developed a protocol for another trial on chronic subdurals, on whether you can give dexamethasone instead of surgery, in fact. But it was dawning on me that these trial designs that we were using for surgery were not a natural fit, particularly for the early stages. And perhaps there was another way to evaluate surgical interventions, particularly those involving technology. Now, I'm not sure why the letters haven't passed, but I then moved on to a PhD in Imperial and to the Hamlin Centre for Robotic Surgery. This is quite a special unit at the time. It was the only research centre where both clinicians and engineers worked side by side. It was led by both Professor the Lord Aradazi clinically and Guangzhong Yang from an engineering side. And it was really formative years for me. And the first thing I did during my PhD was spend a lot of time trying to really understand the approaches. So as anyone who's done a PhD will know, it affords you the opportunity to really think hard about a problem. I developed and validated cadavers and phantoms for these operations to really understand what worked and what didn't work. And when I felt I could do the operation on the cadaver really well and what the problems were, I then tried to use the robots in the market to see if they fix this problem. On your far right is the Da Vinci robot, which is the robot that's most commonly used for minimally invasive surgery around the world. It's a fantastic robot. It has an endoscope, has these arms with articulated wrists, but it does not fit in the hole that you would use in neurosurgery. In fact, one of the arms does not fit in the hole you would use for neurosurgery. So it's completely ill-suited again to the sort of operations in which I was focused. And the pitch, therefore, was to create a mini Da Vinci for brain surgery, something which had the same format. For those of you who have not seen it, the idea of Da Vinci is that the surgeon sits in a console, eating a sandwich while doing the operation, and the robot's next to the patient, able to recreate these movements, removing the tremor, reducing the gain, and giving you dexterity so operations feel much easier. And I wanted to do that for brain surgery so that even I could do these minimally invasive approaches that I'd seen in Zurich safely and effectively. And Guangzhong Yang, the engineer on the far left, and the team of engineers I worked with, Gutier, Chris, and Conrad, spent the next couple of years building these robots for neurosurgery, and it kind of worked. We got not only cadaver studies, but we did live animal studies to great success. And I didn't forget the lesson about evaluating alongside innovation, and my supervisor, Ara, was very insistent on this. And we found that, in fact, people had thought about this problem of evaluating surgical interventions. The Oxford Group for Evidence-Based Medicine, those of you in the audience who are medics and academics will know that there's a very well-established protocol for evaluating drugs, phase 1, 2, 3, and 4 studies. They also, this Oxford group, described something similar for surgery. And that is the ideal collaboration. It's the equivalent for drugs, but for surgery. It allows you to include not just the intervention, but also the learning curve of the surgeon, the change in the patient selection, and things like that. And the group of clinicians I was working with, so that's Tom, Archie, and Richard, who you see pictured, really worked hard with me to try and evaluate these robots using the frameworks from IDEAL. And it sort of worked, but in a more accurate way, it didn't really work. So the robot, as I said, did end up being usable in a cadaver and in a live animal study. So the technical aspect was a victory. But it dawned on me, even as we were doing those animal studies, that there was exactly zero chance this would ever impact patients. Because although it was sort of technically possible, surgeons hated using it. The operations, in fact, which we showed in meetings, were sped up about 20 times because it was so slow to make it look better. Patients, when you surveyed them, also hated the idea of someone operating on their brain sitting away from them in case something went wrong. And lastly, and something I hadn't really considered at all, the economics don't really make sense. The cost to market for a big complex robot for Da Vinci is huge, but a cost to market for an even more complex robot that's doing something even harder is larger still. And so the actual likelihood of it being feasible to build a company and to sell that to surgeons, to use that on patients, it just was a non-starter. Now, I finished my PhD with my tail between my legs and made, to date, the best professional move of my life to Queen Square, which you see pictured here. And it wasn't really abandoning the problem, but it gave me fresh eyes and a fresh start to think about a better way to solve the problem. And the first step was learning to do these operations myself. And I had fantastic mentors, both in Neil Dorwood and Joan Grieve, real master surgeons at doing these operations through very, very narrow holes, and taught me all the good bits about what I know. And seeing these operations firsthand, I could really crystallize the two central problems, which we've touched on earlier. The first is that if you want to do these operations, you actually have to know very well what you're seeing. And that can be much more challenging in these minimally invasive approaches than it is for standard open approaches. The anatomy you're seeing is often much more scarce. It's often covered by bone. And the impact of a mistake you make is so great that it makes surgeons understandably cautious. Now, all of us, I mean, AI is such a buzzword, I hate to say it, but all of us will have experienced this in our day-to-day life. For those of you who use things like ChatGPT, you'll know that almost overnight it's gone from being an accident to not being very good to suddenly being everywhere and pretty good at most things. And the reason things like ChatGPT are very good is twofold. First, because it reads the internet, there's lots of data, and second, because of the compute. But there is no data for surgical videos. In fact, not only is there no data for these operations, most people fail at the very first step of even recording the operation, let alone annotating them. And so the first step in building a ChatGPT for brain surgery, a surgical co-pilot, was to create infrastructure to record everything we did. And so Professor Dan Stornoff, who's on the left, we actually worked together at the Hamlin Center before we both moved to UCL. Worked with a company which is subsequently sold to Medtronic, Touch Surgery, and we used this company's resources to plaster Queen Square with the capability to record operations and to store them so that we would keep all that data and be able to use that to better help patients in the future. At the same time, I rethought about how to solve this problem of executing these movements. And actually, if you look at keyhole operations, the tricky bit about doing the operation is not that it's a small space, but that your arms are tied together, that you're going through this long, narrow corridor. And the reason it's tricky is because if you're working down a long, narrow corridor, you can't triangulate, and without wrists, you can't do complicated things like stitching or dissecting or cutting. You end up just poking and pulling, doing coaxial movements. And so to actually, although the Da Vinci is a really impressive robot, the actual helpful bit of the Da Vinci is not the 1-ton, £1 million machine. It's just the wristed instruments. That's the actual useful bit of the operation that we need help with. And so we reverse engineered it, and in fact, we thought a better solution would be to have a standard instrument, but just with the wrists, and to try and remove everything else. And so doing a handheld robotic design we thought would be the way to go. And the National Brain Appeal, who provided this initial seed funding without which we would not be able to do this work, was hugely instrumental. I'm proud to be a trustee of an MBA now, but without it, we wouldn't have been able to start it. And lastly, I had touched on the ideal collaboration, and it was the only framework developed for evaluating surgical innovation, but even the ideal frameworks didn't cover technology, didn't cover what to do in the lab at those first steps. It wouldn't have stopped me making the mistakes I did at Imperial building a very complex robot to fix a small problem. So I worked with Peter McCulloch, who's the chair of IDEAL, and I asked him, if you don't have this, can I create one with you? To which he agreed. And we then developed this framework to evaluate surgical instruments and innovations at the very early stages of the lab. The idea being that we could then save so much effort by making the right choices early on. At the end of my training, I had the extraordinary good fortune of having both Mary Murphy as a clinical director and Rob Brownstone as the academic lead, who were just incredibly supportive of me and broke their back, I think, to create me a job for which I'm eternally grateful. I also had, as an icing on the cake, a consultant appointment at the same time as my best friend during training, Patrick Grover, who's the new clinical director and at the back there. And what made Patrick special was that he was not only an excellent surgeon, but also a very good guy. So I'm very lucky to still be working with him now. At the same time, we had some catching up in my life. These pictures are all within 12 months of each other. So I completed my training, did my last exam, became a consultant, got married, moved house, had a kid, and COVID hit, all within the space of 12 months. It's a busy year. My son, he's at the very front, and slightly embarrassed at me calling him out. It was a real impactful part of my life. Now, with my consultant appointment, I then managed to continue to push these various strands forward. The clinical work doing pituitary surgery is inherently multidisciplinary, and you can't do it just as a surgeon. You have to work with outstanding endocrinologists, lots of whom are in the room, and ophthalmologists and oncologists and pathologists, a whole team. For the surgery itself, it requires not just the surgeon, but also fantastic anaesthetists. Again, I can see some in the room, and nurse specialists. They all really help us do these operations and manage the patients after the surgery well. But taking our eyes beyond just the patients we treat in front of us, I worked to either create societies, in the case of the British Neuroendoscopy Society, but also take leadership roles in existing societies, the Pituitary Society and the Pituitary Foundation, to better come up with ways, frameworks, guidelines to help patients with pituitary disease. And this had the unintended but fantastic benefit of helping my AI research, because I talked earlier about the need for AI to have lots of data. And once you have this library of 1,000 videos that we do now, that we did back then, the next step was trying to make sense of it. You can't just give all the videos to AI and say, here's the operation. You have to teach it. You have to teach it what the anatomy means, what the steps are, what the instruments are. And then you have to ask more basic things. What is a good operation? What's a bad operation? What do those outcomes look like? Now, this may seem very obvious, but no one ever wrote this stuff down. There's not one description of what a normal pituitary operation should look like, what are the variations in that operation, and nor was there any patient-reported outcome measure for pituitary surgery, nor was there any core outcome set from pituitary surgery. We created all those things and led that work here at Queen's Square, and that's the absolute foundation for the AI work, because we then were able to annotate this huge army of patient data with this structure that we developed working with patients and other endocrinologists and surgeons. And with all that out of the way, we could then work to build these AI co-pilots. Now, Adrito, who I saw earlier, I don't know where he's run off to, is our PhD student. So you can see in the very center, myself and Dan Stoynoff, who I showed you earlier, and Adrito on the day he passed and became a doctor. And Sophia on the left and Evans on the right are the associate professors who drive this with us. And Adrito, for his PhD, used all this data to create the models, to describe the anatomy, to predict the steps, to predict the next steps. And in the culmination of this work, we have now taken this just a couple of weeks ago to patients. So the first AI to be used in live surgery for the brain anywhere in the world was here in Queen's Square just a couple of weeks ago, in fact. The people who led that work are Johar Mar at the top, the engineer, and Dan Khan at the bottom. And it worked exactly as we'd hoped. It's a very first step. So you'll see this is doing simple things, recognizing basic anatomy. Some surgeons in the room will say, I could have told you that. But it's the first step. And anyone, again, who's seen AI in everyday life will know that actually the difference between doing it a little bit and suddenly being better than everyone else is a small difference. It's just data. So I'm really proud of this work. It's a first step, but it's a great step. And the robotics side, I had, again, the incredible luck of picking a PhD student, Dan and I supervised, who's much better than myself, Manios Dimitrakakis, who's also here with his team. And Manios, as an engineer proper, built a much better robot than I did during my PhD. He built a handheld platform that was able to solve this problem of articulation, even at the sizes required for neurosurgery. No one had done it in the world to that point, and keep it in a handheld form. That work later span out to Panda Surgical. We have a group of engineers, in fact, another to add soon, who again are here. And these engineers are driving the development of this robot to bring it to patients. And we have already, in fact, used this robot in cadavers with John and Joachim, who are here, again, pushing forward with that work. And we've also used it in live animal studies. It's slightly cringe watching videos of yourself, but the point of this video is to show you what it looks like in the hands of a surgeon. It looks like any other instruments. You can change the tips in seconds, and the tips of the instruments have this all-important wrist motion, which gives you the ability to do things like cut, dissect, and stitch. And we're raising, so if anyone has a spare million. And lastly, we moved to create frameworks to evaluate these technologies. So in our role in IDEAL, my team of clinical research fellows, again, you can see them pictured there, Simon and John, Dan and Hugo, created new frameworks that didn't exist to evaluate both AI and robotics in surgery. And I list them here for the paper just to show that actually they did the hard work and to recognize that. But actually this has been adopted very widely. Lots of robotic platforms now use the IDEAL framework, ourselves included. So in the last few minutes, I'd like to turn to what comes next. And perhaps the first prediction is obvious. We really want to help as many patients as possible. So we want to bring our technology, both the AI to help surgeons make better decisions and the robotics to help surgeons execute those decisions more faithfully, to as many operating theatres as possible, in as many hands as possible, to do as many operations as possible. That's the ambition. And it won't be what you just saw. It won't be AI that draws around a structure. It will be AI that you talk to like an expert in the room, asking them, what would you do next? Do you think I've taken enough? Should I stop? Similarly for the robots. I'm very proud of the robots. The first in human studies for that robot should be next month, which will be a huge milestone, but that's the beginning, not the end. The goal is a suite of instruments, not just that enhance your dexterity, but perhaps another instrument that enhances your precision, or another that might tell you when you're applying too much force. And this instrument portfolio is what I think will improve surgical performance. And we have a network of surgeons around the world that we closely collaborate with, and we've also in-house had these fantastic co-investigators. Patrick, I mentioned earlier, who's an aneurysm surgeon, as well as a scar-based surgeon, is using the AI we've developed in his work. We have George and Parag for endoscopic spine surgery, using both the robotic and the AI work in endoscopic spine surgery, as well as for meningioma surgery, glioma surgery, open surgery like insertion of ICP bolts, even epilepsy surgery. The next prediction is around not just doing better operations, but doing new operations. And Will Muirhead, who you can see on the far left, pictured in blue scrubs, was my first fellow who actually approached me while I was still a trainee myself and asked if he could do research with me together. And our plan, both Will and myself, was to do research looking at either robotics or AI or simulation. And he did all of those things, but decided they were a little bit boring. And then had this idea to, in fact, use technology to do something even more ambitious, which is to interrogate the brain itself. Now, on the left, the person next to Will is not me, but my handsome colleague, Harith. And they're using Neuralink in patients. But I actually think the most exciting way to use technology is not to just use what others have developed, but to develop stuff ourselves in-house. And Hugo, who's pictured in the center, who's one of Will and I's PhD students, is using NeuroPixel technology, which is a probe where UCL contributed to development, to interrogate the brain and hopefully in the future give us new treatments we can't yet offer. And to do these new operations, we need new, I would argue, better surgeons. And we have, as a part of our ambition to create these simulators to validate our robotics and AI work, this huge array of simulators now that we've used to train surgeons better. Austin Coe, who's pictured on the far left, has driven that work in spine surgery in particular. And those simulators have formed the foundation of our Queen Square Simulation Centre, where 2,000 people now have passed through the simulation centre in the last 5 years, a huge achievement even in its own right. And our MRES in neurosurgery, with Mo Kamal and myself there celebrating 5 years just recently, and some of those MRES students are here today. But of course, what I've outlined is really still the same. It's those same 3 strands: doing surgery, using technology, and trying to evaluate as you go along to make sure we're doing the right thing. So technology changes, the purpose really does not. And I think with that, I'll thank everyone for their attention, thank the many people in our group and our many collaborators and funders. Thank you very much.
It's my turn to deliver the vote of thanks now to Hani. And I didn't realize that what STEM stood for, but surgery, technology, evaluation, Marcus. So that's when they're always talking about STEM, they're talking about you, Hani. My name's Rob Brownstone. I'm the head of the Division of Neurosurgery at the UCL Queen Square Institute of Neurology, which is where we are now. I'd like to welcome you all and say good evening. It's a real honor and privilege that Hani asked me to deliver the vote of thanks. I come from Canada. In Canada, becoming a professor is no big deal. I don't think anybody noticed when I became a professor. It was just absolutely nothing. Nothing happened. I don't even think I told my mother, in fact. And here we have the whole family here. It's fantastic. So it's a bit odd, this tradition, but it's really fantastic. Because becoming a professor here in this society is very well recognized across society, and it's a real badge that you wear of intellectual distinction. And as you've seen in this lecture, Hani has really earned this title through years of hard work and sustained achievements toward bettering the lives of our patients. That has been his sole focus, clearly derived from his parents, and he has continued that family tradition. I can truthfully say that when he finished his training, we were super lucky to have recruited him. I felt at the time that we were lucky then, and I've constantly been reminded of that fact over the years, and again this afternoon when we were all reminded of his tremendous accomplishments. I've come to know Hani in several different spheres over the years, first as a trainee, then as an early career researcher, neurosurgeon, and now as professor. And in addition to the academic sphere, we interact regularly, in fact, every second Friday in the operating theater. Since we share a room on that day, we begin the day joining each other in what's called team brief. And so for the non-surgeons here, team brief is something that's designed really so that you can delay starting the first operation of the day. But it starts off by everybody standing in a circle and introducing themselves to each other as if we haven't worked with each other before. And we go around the circle, usually 10 or 12 people in the room at the time. And on about 2 occasions in the last couple of years, we've gone anti-clockwise when we've introduced ourselves. And I didn't even notice, because I don't notice which way we go, but that really unsettled Hani. And what was remarkable about that, I did feel really badly from him, but what was remarkable about that was not that it unsettled Hani, but in the way he recovered from it. Because really, when something unsettles him, whether it's something as trivial as team brief going around the wrong way, or something far more significant, he adapts quickly, he stays calm, and he keeps his attention focused on what's really important and what really matters. And he would show that then. And that's really one of the qualities that makes him such an outstanding surgeon, colleague, and leader. This afternoon, we've heard about remarkable scientific advances, his robotics and his use of AI, and the impact that they're already having on patient care. Behind all of that is the same combination of curiosity, resilience, and determination that we've been fortunate enough to see every day around here. So please join me once again in thanking Hani and his parents for being such fantastic role models, but not only for a brilliant inaugural lecture, but for everything he's already contributed to our patients, to our trainees, many of whom are here today, to our profession, and for what I'm sure will be many more years of outstanding achievements. Thank you, Hani.
Okay, thank you everyone. We will now move on to our second inaugural lecture of the evening, and it's really my great pleasure to introduce my departmental colleague, Professor Rachael Scahill. So Rachael has promised us a non-linear journey, and already in preparing this introduction I learned something I didn't know, which is that Rachael's first degree was in zoology from Oxford University. So I'm really excited to hear how we got from zoology to Huntington's disease. After Oxford, she did a master's in computing and then moved here to undertake her PhD at the Dementia Research Centre, where she first began to work on the application of MRI to study familial Alzheimer's disease. But since 2008, she's worked alongside Professor Sarah Tabrizi, leading the image analysis team on Track HD and the young adult study, so shifting her focus to Huntington's disease. Rachael's research interests are in the development of novel tools for the analysis of neuroimaging scans to further our understanding of the natural history of Huntington's disease, and as well as helping to develop biomarkers for use in clinical trials. She's the Associate Director of the UCL HD Centre and the co-chair of the European Huntington's Disease Network Imaging Group. And as a departmental colleague, I can say it's really a pleasure to work with Rachael. She's very modest, and also this doesn't cover all the work she's done to support students in our department, and she's just a wonderful colleague. So please join me in welcoming Professor Scahill to deliver her inaugural lecture.
Thanks, Selina, and thank you all for coming to hear me talk about my journey. I've had a few deviations along the way that I'm going to talk you through. So I'm really fortunate in that I can really remember when I first became enthused about science. So it was not in a classroom, but actually walking up Helvellyn in the Lake District on a family walking holiday, when I was obviously being a little bit of a pain in the neck. I was about 10 years old, having a moan, and Mum decided to distract me with, of all topics, the genetics of eye colour inheritance. Which seemed to work. So this is my mum. Unfortunately, she can't be here today, but she is hopefully listening online. So this wasn't a totally random topic for Mum. She was a natural sciences graduate from Cambridge in the 1950s, back when not many women did that sort of thing. And she subsequently became a teacher and always had a real enthusiasm for sharing her knowledge, and having taught in a deprived area of Liverpool, as well as volunteering in a Young Offenders Institute, she's got a real passion and understanding of the value of education for all. But she got me hooked in science. So I was working hard at school. I set my heart on going to Oxford University, following in the footsteps of my brother and my sister who are here, and everything seemed to be going very well. I was then on another walking holiday in the Lake District, the site of many exciting life events when I encountered potential deviation number 1, which was Aidan, as I now know. So I was a 17-year-old schoolgirl in Liverpool. He was a 19-year-old trombone player in the Scots Guards Band in London. And I think there was some concern among the family that I may be going potentially off track. But I have to say, there is nobody more supportive and more determined that I should achieve my aim than Aidan. So I was fortunate, and I did get a place at Oxford to study zoology. I had fantastic friends there, some of whom are here today, and I really loved my subject. I was very fortunate that I loved all the study until my 3rd year research project. So my tutor was an entomologist and thought that it would be great for my project to learn about the feeding habits of locusts. Not in an exotic location, unfortunately, but in the zoology labs in Oxford. And frankly, I just didn't get on very well with the locusts, and they seemed to show a sort of willful disregard for my hypothesis, and made me really rethink whether science was for me, whether research was for me. So I did the next obvious best thing, which was to become a manager with W.H. Smith's. I did that for around 5 years before I thought I just really miss science. And so I went home to Aidan, who we were married by this stage. I said, I think I've made a mistake, and maybe rejecting science just because of some badly behaved locusts was a bit rash. So Aidan, as always, was supportive and said that if I wanted to go back and do a master's to get back into science, then he would support me in that. So that's what I did. I did a master's in scientific computing, and at the end of that, I was obviously looking for a job, and I saw a small advert in the Evening Standard, back when we used to look for jobs in the local paper, and it was advertising a job for looking at MRI in Alzheimer's disease and working with Dr. Nick Fox, as he was then. So I loved the idea of this job, was very enthusiastic, and was fortunate enough to get an interview. The night before my interview, I'm cramming to learn everything you could possibly know about Alzheimer's disease and read all Nick's papers, when I got a phone call. So Aidan at this stage was touring the States with the band, and the phone call was from New York. They said that he'd been taken into hospital and had an appendicitis. There was a chance that they might need me to fly out there to escort him back, because obviously he was on a tour, the rest of the band were moving on. So that was a possibility. So obviously I was very worried, because I really, really wanted this job, and I did not want to miss the interview. Fortunately, the army managed to arrange an escort for him to come home, and I did. Not because I was going to the interview; they did that anyway. But so I went along to the interview. Really loved the idea of the job. Felt I'd done quite well in the interview. So when I got a phone call from Nick Fox a couple of days later, I was sort of devastated when he said, "I've given the job to somebody else." But he said, "I might be able to find you something." I subsequently found out that I was referred to as the wild card, and a job was indeed created for me, for which I will always be eternally grateful. So I started my work at the Dementia Research Centre, and just to give you now a bit of background about Alzheimer's disease. So it's a neurodegenerative disease, the most common cause of dementia. Usually starts in old age and results, as I'm sure we're all familiar with, progressive memory loss. And it's caused by a buildup of amyloid plaques and fibrillary tangles, which leads ultimately to neuronal dysfunction and death. In the very rare cases, it can be inherited in an autosomal dominant form. So that means that if you have an affected parent, you have a 50% chance of inheriting the disease. So that's familial Alzheimer's disease. And that typically manifests much earlier and those at risk of developing the disease can undergo a predictive genetic test. And the advantage of when people have this test, this allows us to study the disease from the very earliest stages. Before they're showing any symptoms, we can look at the pathological process from the very earliest point. So this is to illustrate post-mortem brains. So on the left, we have one from Alzheimer's disease, and on the right, a control who did not die of a degenerative disease. And you can see the devastation that the pathology has caused in this brain. But this is, of course, end-stage disease, and what we want to know is what's the process, how did we get there? And this is where imaging comes in. So on the left, we have a normal healthy control, and on the right we have a scan showing Alzheimer's disease. And if you focus on these areas, the hippocampi, that we know, it's a structure we know is associated with memory, and this is really devastated by the pathology. It starts there and eventually spreads to other parts of the brain. So that's in fairly well established Alzheimer's disease. But earlier on, it may be more difficult to tell the difference between two scans if we're just looking at one scan. So on the left, we actually have a normal healthy control, somebody who happens to have slightly enlarged ventricles. These are the fluid-filled spaces in the middle of the brain. And this is somebody with very early Alzheimer's disease. So just looking at a single scan may not be enough to tell you whether somebody's got Alzheimer's disease. So I'm now going to talk about the brain boundary shift integral, which is a technique developed by Nick Fox. And as I said, if we have a single scan from an individual, we may not be able to tell the difference, but what is really powerful is to look at the change over time within an individual. So you can measure the brain at time 1, measure the brain at time 2, and subtract, but there's measurement error associated with both of those things. So I'm now going to shamelessly use Nick's fantastic analogy, which is when you're measuring two things, it's a bit like when you've got two kids in a playground. They don't measure one person and then the other to say who's bigger. They stand side by side. So at this point, I'll introduce my children. We have Patrick and Hope. This is Hope's first day at secondary school. So she's got her big brother here to protect her, who is nearly 2 years older. But you can see with them lined up, you can see that difference in volume, if you like, and that's the most sensitive way to pick up the change within the brain, for example. So that is your first scan. So people go into the scanner in different positions, but we can get the computer to align the images, and that allows us to look at the change that happens over time. So that is baseline scan, and that is 2 years later. So you can literally see the pathology progressing there. And if we look at the focus on the hippocampi, they are shrinking down over time. We can quantify that with the boundary shift integral, so we can get a number out, see how much the brain is changing. And this is the paper that Nick published in The Lancet back in 1996. And if we look here, we have the controls, and then there is good separation between people with controls and people with Alzheimer's. So this is a really powerful technique for looking at change over time within the individual. We have at-risk people here, so these are people who have had a genetic test but aren't yet showing symptoms, but already have an elevated loss of volume, a higher atrophy score. So as the boundary shift integral implies, you see change at the boundary. You can see that moving as we flick between the scans. But that's not necessarily where the tissue losses come from. So this is, we get this through what we call a linear registration. So we're treating every point within the image exactly the same. We might rotate a bit, move left or whatever, and get that match between the second scan and the first. If instead of that we do a non-linear registration, and we can say the voxels within the image can do whatever is needed to get a perfect match between the second scan and the first. So we get rid of all the change and get that perfect match. We can then interrogate what did it have to do to get there, and we get what's called a voxel compression map through this nonlinear registration. And we can now see that, so here we've got blue and green means contraction, red and yellow means expansion. I don't know whether it shows up very well with the lights on, but we've got expansion of the fluid-filled spaces because there's loss of tissue, and that tissue seems to mainly come from the temporal lobe, which is what we would expect in Alzheimer's disease. We can apply this technique to other forms of dementia. So frontotemporal dementia, and this is really quite striking, how localised the change is to the temporal lobes here. This is CJD, which we know is movement disorder, and here the pathology is very much focused in the cerebellum. So these are longitudinal scans from a single individual, and so in a way this can be a little bit anecdotal. What we really want to know is where are there robust, statistically significant differences between a group of people with, say, Alzheimer's disease and controls, so that we can reliably identify the regions associated that we might want to potentially target with treatment. So this is where something called voxel-based morphometry comes in. So everyone, as I say, it goes into the scanner in different positions. What we need to do is spatially align them all so that we then have a point in the image corresponds to exactly the same anatomical location in all the different people. So we put them all into the same spatial framework, and then we can do statistics to say where are there significant differences in a particular group. So this is work done with John Schott, and here we can see that in the pre-symptomatic people, so these are people who've had a positive genetic test but aren't showing symptoms of the disease, the pathology is very much localised to the hippocampus, this area here. When we go to mild Alzheimer's disease, it's become a little bit more spread out, and by the time you have moderate signs of the disease, the pathology is much more widespread. So this gives us an idea of the sort of evolution of the pathology over time. Another thing I did whilst at the Dementia Research Centre was look at different mutations. So I said in familial Alzheimer's disease, there's a genetic mutation that causes it, but there are different forms of the genetic mutation. And here I wanted to look at what is the pattern in people with an APP mutation, and this is the pattern in another form of the disease, presenilin 1. So these people are matched in terms of the severity of their disease, but they have a very different pattern of pathology and much more extensive in presenilin 1. This was work I did quite a while ago with Jed Ridgway. So I'd been at the Dementia Research Centre for quite a while. I did a PhD and then did an MRC-funded postdoc. And I really felt like I'd become quite narrow. So I was using one imaging technique in one particular form, one sort of neurodegenerative disease, and I really wanted to broaden out. So I saw a job advertised at the British Medical Journal. And this was developing an evidence-based tool for clinicians to use in hospitals. And I thought this sounded just the sort of thing to broaden out. I got the job, but it became apparent quite soon that this was maybe not the smart move. So it was little more than glorified telesales, really. I was ringing up very busy doctors and offering them very little money and very little time to write an article. And those of you who know me know sales is probably not really my thing. So it was not very successful. So again, I go home and say to Aidan, I think I've made another mistake. This is not what I hoped it would be. Aidan at this point has left the band and has retrained as a teacher. So in true primary school fashion, he said, no, there's no such thing as mistake, merely a learning opportunity. So, but I thought I maybe had burnt my bridges by this stage. How do I get back into research? And this is when, lo, an angel appeared in the form of Sarah Tabrizi. Thank you very much to Mina Farag for this, for the doctoring of this picture. But Sarah rescued me. So she was working on a new project and she needed somebody to lead the imaging team. I joined what subsequently became the HD Centre, and to be honest, since then, I've never looked back. So just to give you a bit of a background about Huntington's disease, it's an inherited neurodegenerative disorder, and there are really three features of it. People have progressive motor impairment, cognitive decline, and neuropsychiatric dysfunction. As with familial AD, those who've got an affected parent have a 50% chance of developing the disease, and they can undergo a predictive genetic test from the age of 18 to find out whether they're going to get the disease. So studying these gene carriers can really help us map the disease progression from very early adulthood through to established disease. This may help us identify therapeutic targets for future clinical trials, and also we can develop markers of disease that may give us an indication when we have a treatment whether it's actually having an impact on that natural disease progression. So the situation at the moment in Huntington's disease is that there are many promising treatments on the horizon. They are being trialled in cohorts of people who are already showing signs of the disease. So in terms of the pathology, Huntington's disease is caused by a CAG expansion in the DNA. We all have a certain stretch of CAG expansion here. If it is between 6 and 35, generally people are considered to not be affected by the disease. If you have 40 or over repeats, then in the normal lifespan you can expect to develop Huntington's disease. 36 to 39 is a bit of an intermediate area, and people have an increased risk of developing Huntington's disease and may even have an increased risk of passing it on to the next generation, because we know that CAG can increase over time. So this expansion of the CAG leads to formation of an abnormal protein, and ultimately this affects the cells. There is dysfunction and cell death. And the longer the CAG, the earlier you would expect to get the disease. And there are some cases where people have expansions in the hundreds, and they would get Huntington's disease as a juvenile. But that is exceptionally rare. So the study that Sarah recruited me to work on was called TRACK-HD. This was an international study. We had centres in London, Leiden, Paris and Vancouver. And although there weren't really any treatments at that time, we wanted to be ready so that when they came along, we knew what markers we should use. We wanted to test them. Are they the same in Vancouver and in Paris, etc.? We had a whole array of tests. We had MRI, which is what I worked on, but we had cognitive testing, clinical testing, blood samples, etc. And they had annual assessments from 2008 to 2011. So this is the motor task. So typically we assess Huntington's disease by a clinician looking at a patient and scoring how their movement problems might be. But the idea behind TRACK was that we could get some more objective measures. So we had an eye tracker, we had a transducer that would measure people's tapping, we had rather horrible tongue protrusion force, but this was much more objective measure of motor impairment than we'd previously been able to have. So when we looked at how much people had changed over 12 months in this case, we found that, so this is the control group. We have pre-manifest A. These are people far from onset, so more than 10 years from onset, between 10 and 15 years from onset. Pre-B is closer to onset, within 10 years, and then we have stage 1 and stage 2 Huntington's disease. And we can predict when people are likely to get the disease by looking at their age and CAG, and we can put that into a calculation and come up with an estimate. So we're only seeing impairment in motor function in the stage 1 and stage 2 people. There's really no signal in the pre-manifest phase. This is a single-digit modality test. This is really widely used. And if you think of this in terms of, it's an international study, you really need a measure that people from different countries can use. And this is a sort of decoding test where you would look at the symbol and put the appropriate number in the box. And we found that there, again, we're just getting impairment in stage 1 and stage 2 Huntington's disease. There's insensitivity in the pre-manifest phase. So ultimately, we want to be able to treat earlier when there's not too much damage in the brain. But we're going to need a marker. We're going to need something to measure to determine whether a treatment's having an impact. So this is where imaging shows some promise. And here we had 4 scanners, London, Paris, Leiden and Vancouver, and 2 different sorts of scanners. So the top is a Siemens scanner, the bottom is a Philips scanner. And there are subtle differences in grey-white contrast. And we really wanted to check how can we get reliable measures at all these different scanners to come up with reliable measure of change over time. This is a baseline scan from somebody who is more than 10 years from onset. This is after 12 months. And you can see the shrinking down of this structure here, the caudate. So that's 12 months, 24 months. Again, you can see this pathology in action. And when we quantify that, we find that whole brain, suddenly now, we've got sensitivity in that pre-manifest B period. So within 10 years of onset, you have a greater loss of brain than a normal control. If we just focus in on the caudate, actually now we have sensitivity from the very earliest stage. So more than 10 years from onset, your caudate is losing volume faster than in matched controls. What's important to people with Huntington's disease, though, is not necessarily what's going on in their brain, but how it's affecting function. So we need to try and demonstrate that there is an association between what's going on in your brain and the function, because if we're going to give a treatment, we want to know that that's going to translate, hopefully, to a clinical benefit. The truth is you're never going to know until you start a clinical trial, but trying to demonstrate an association goes some way to suggest that this might have an impact on function. So this is a paper that I did a while ago demonstrating that there was an association between impairment on that tapping task and loss of grey matter in the caudate and putamen. Another question we asked was, can we identify those who progress and those who don't? So, as I said, we can estimate when people are likely to get Huntington's disease by looking at their age and CAG. But there's still a reasonable amount of variability around that estimate. And some people progress faster than others. And we found that looking at whole brain, caudate, grey and white matter was a good predictor of who was going to subsequently progress in terms of function. And some of the early signs were speeded tapping and also recognition of negative emotions. So this is something that people might not necessarily feel themselves or even the partner that they live with, but this impairment happens really early on and can actually start to affect relationships even before people have got a clinical diagnosis of Huntington's disease. So Track HD, this study, together with other large natural history databases, the Enroll HD and the Predict HD study, allowed us to build up this idea of what was going on in the disease from 15 years before. So mostly everything is normal, but we know that from the earliest stages, there's already loss of the striatum, that's the caudate and putamen that I was talking about before, and there are elevations in certain proteins. One key one is neurofilament light, or NFL, and also mutant huntingtin, which is a sort of toxic element that contributes to the pathology. But, of course, we want to know what happens earlier on. Can we identify the start of that process? Because that's really probably where we want to intervene. So that's when the young adult study was born, and the aims were to give really deep phenotyping of a cohort in the furthest from onset previously studied in terms of adults. And we wanted to establish sort of disease markers when they first become detectable, and could we potentially use those as endpoints in future clinical trials? And then can we identify that window? So can we find the time when you get the start of that pathology, but everyone is clinically well? That's when we really want to give a treatment. So we had 64 gene carriers who were around about 24 years from onset. We had matched controls. We did a whole range of different imaging assessments, biofluids, cognitive neuropsychiatric function, and essentially there were no difference in the majority of the measures. So remembering that this is 24 years from onset. So this is really good news for the participants. There is we can't tell the difference between controls and people who carry the gene in terms of their cognitive and neuropsychiatric function. But there are subtle signs very early on in reduced volume in the caudate, but also elevations of some of the biofluids, for example, neurofilament light. And we didn't just look at blood, we also looked at CSF, so the fluid that bathes the brain. We collect that through a lumbar puncture, and that gives us really detailed information about the proteins that are within the brain. So that allowed us to build up this picture and track back. So actually it looks like NFL and mutant huntingtin start to deviate round about 25 years before onset, but there's still no cognitive or motor impairment at this stage. The striatum is smaller from the earliest time we've looked, and we really don't know whether that might even be a neurodevelopmental effect. People who've got Huntington's disease have had the gene from birth, so we don't know whether it never develops normally or it develops normally and then starts declining. And there is work going on in the States looking at-risk children to try and establish whether development is normal in people carrying the gene. So I just wanted to mention at this point the Huntington's staging system. So this was developed by Sarah to try and give a framework for the disease. So looking forward to clinical trials, much like you get staging in cancer, you want to know what point people are at so that you can maybe identify people who are going to be appropriate for a particular clinical trial. So, as I say, everyone with Huntington's disease has had the gene from birth. So if you have 40 or more CAG repeats, you are already stage 0. To transition to stage 1, you need to have a reduction in your caudate and or putamen volume so that you are then outside the control range. Stage 2 represents very early subtle impairment, an elevated total motor score and or impairment on the symbol digit modalities test. That's the one that I showed you before. But people at this stage do not fulfill the criteria for Huntington's disease. So it's really only very subtle change. And then stage 3 is where they can get a clinical diagnosis of Huntington's disease, and this is where it's really starting to affect their day-to-day life. But that is an incredibly useful framework within which to work when we're recruiting for clinical trials. So we then followed up the HD-YAS cohort 4.5 years later, and interestingly, as you would expect, we still saw the volumetric difference and the biofluid difference, but now we start to see slight change in brain microstructure. So the white matter connections within the brain are already starting to break down. And this is people now 20 years from onset, but still crucially, clinically, absolutely fine, indistinguishable from controls. So this is showing here the putamen atrophy. This is somebody at time 1 and time 2, so all the lines are joining up the individuals. We can now do this within the staging system. This is somebody with stage 2, so they start with a smaller putamen and they are declining. So the interesting thing is there's actually, in terms of volume, still quite a lot of overlap between the controls and people with Huntington's disease, but pretty much everyone is declining from that very, very early stage. We also see this effect in CSF neurofilament light. So the controls is really very little going on, but pretty much everyone with Huntington's disease has elevating their NFL over time, and here we have our stage 2 person right at the top. And another really interesting discovery with this paper was that there was what's called somatic expansion, which we could find was a driver of the disease. So I said that people inherit, say, 40, 41 CAG repeats, but within the cells, we also get what we call somatic expansion, and so the CAG gets larger and larger, particularly in vulnerable cells within the brain. And it seems that they hit a certain threshold before the pathology starts to affect that cell and it starts to break down. So this somatic expansion, which we measured in blood, was a really strong predictor of change in caudate and putamen, and also NFL. And PENG is a measure of sort of the integrity of the caudate and putamen. So this was an individual transitioning from stage 0 to 1. And as you would expect, because that stage is defined by a loss of the caudate, we can pick it up very nicely in the imaging. We can measure it with the boundary shift integral, and we can do this colour overlay, the voxel compression map, and you can see it's really, really localised at this stage. It's not affecting other areas of the brain at this point. If we look at the staging system, this was done by Harry Knights. If we look at stage 1, the vast majority of our cohort were in stage 0, so not showing any signs of striatal loss at this point. But at visit 2, 10 had moved from stage 0 to stage 1. We didn't have any more stage 2, and none regressed, which is sort of makes sense. You don't expect people to go backwards, but that is a sort of validation of the technique that that nobody moved backwards. So just to summarise, in this paper I did together with Mina Farag, and it seems like we've now found this treatment window where we would like to intervene, where there's normal function, but we've got detectable biomarkers showing subtle early change, and this is really promising for taking this forward for prevention trials. Mina then subsequently went on and correlated this, the biofluids, so on the top we've got NFL, and on the bottom we've got NFL, top we've got PENK. So PENK is very localised, but NFL is much more widespread. So now I've saved the best to last, technical developments. So you may not think this sounds very exciting, but this is, to me, important for 2 really important reasons. Firstly, of course we all want to get our name on a paper that says we've cured Huntington's disease, but we do not get there without absolutely obsessional attention to detail for the techniques that we use. Secondly, these small technical projects are absolutely ideal for masters and PhD students to own a project, to get a first author paper, and to learn about the rigours of scientific research. So I have been blessed with many, many fantastic students, several of who are here today, and I'm just going to talk you through a few of the projects that they've worked on. So Jo Barnes, Professor Jo Barnes, so she is the person to go for all things hippocampal. She may not be aware, but she is on 60 papers that mention hippocampus in the abstract. So she took Nick's whole brain BSI and applied it to the hippocampus. So we know the hippocampus is very vulnerable early on in Alzheimer's disease, and maybe we can get more sensitivity just by focusing on this area, but it takes a lot of adaptation. What she needed to do, traditionally we sort of draw around the hippocampus, and I had many, wouldn't say necessarily many happy hours, but many hours in my PhD literally drawing around the hippocampus. But it takes an hour per hippocampus. We've got a left and a right one, and we have many hundreds of people with scans, particularly if we're talking about clinical trials. It's just not feasible to do this manually. So Jo did a real rigorous examination of automated hippocampal BSI to say, can we get it to look as if we're drawn around it manually? And indeed, yes, we can. So why is this important? Well, as I say, it's just not feasible for clinical trials that we're all drawing around hippocampi, and we would probably all go mad. But so this tool is now adopted in clinical trials worldwide. So this is actually having a real impact. So then I'm just going to talk about Nikki Hobbs' work. So she took the boundary shift integral and applied it to the caudate, a different set of challenges to applying it to the hippocampus. And so she demonstrated that volume loss measured manually versus the BSI, we get very good agreement. Now here we have a Bland-Altman plot. Nobody loves a Bland-Altman plot more than Nikki Hobbs. She loves them. And you don't need to worry about the detail, but essentially, in terms of a Bland-Altman plot, this is good, this or this is bad. Okay, remember that. So this is good. She's got really good agreement between automated techniques and manually. Again, why is this important? Well, we can now apply this in clinical trials in Huntington's disease without having to draw around the caudate, saving many, many hours, and important for pharma companies, saving lots of money. Then I just want to talk about some work by Susie Henley. So I mentioned voxel-based morphometry, which it can be a bit of a black box. There's lots of parameters to choose, lots of different buttons you can press, and depending on what button press you do, you get different answers. So Susie did a really rigorous interrogation of how does parameter choice affect the results you get. And this is just one example where she looked at the level of smoothing in the scans. And you can see that you do get different answers. You get different regions affected. So this is going to affect our interpretation of the disease. So this is really critical work. I then wanted to mention Annabelle. So Annabelle came to us as a master's student several years ago, and delighted that she stayed on to do a PhD. And what she's working on at the moment is scan harmonisation. So there are literally thousands of scans from people with Huntington's disease around the world. What we'd really like to do is pool them. We get incredible power from pooling all these images, but that's not easy to do. Different scanners come up with different things. There's different grey-white contrast. So she's done a lot. This is the sort of intensity profile before and after harmonisation, and she's getting really good results from this so that we can join all those scans together. And why this is important is obviously the more scans we've got, the more we can understand Huntington's disease, but there are particularly rare forms where there's an additional sort of alteration in the DNA, atypically, which can change the age at which you get it by up to 15 years. But this is so rare that we're really only going to understand what it's doing to the brain by combining large data sets. And finally, we have Harry Knights. So as I mentioned, he worked on the staging system. And to transition from stage 0 to stage 1, you need to have a smaller caudate or putamen. It is measured with software called FreeSurfer, version 6. And Harry wanted to ask, okay, so what happens if we use version 7? We would hope that it would produce very similar results, but unfortunately not. So this is, excitingly, a Bland-Altman plot. It does this. This is bad. So not only do we get differences between whether you use version 6 or version 7, but the magnitude of the difference varies according to how big the structure was in the first place. You don't need to necessarily get your head around that, but this is bias upon bias, which is thoroughly bad news. Maybe slightly obsessed, Harry then moved on to what if we use a different pipeline? So there is a pipeline for whether you've got one scan for individual and a different pipeline if you've got more than one scan, the longitudinal pipeline. And indeed, this affects the staging. So we get a different proportion of people in stage 0 or 1 depending on which of those pipelines you use. So why is this important? Well, we are now starting to recruit people on the base for clinical trials on the basis of their stage, and what we really want to do is go as early as possible and identify, for example, stage 1. But if whether you get in or out of that stage is critically dependent on where you put this threshold, and if there are different things putting you one side or other of the line, then that's going to affect whether you get into a trial or not. And really importantly, we're now using stage transition, or proposing to use this as an outcome measure in a clinical trial. So we may even decide whether a drug is good or not, depending on whether you've crossed that line. So we just really need to understand what influences where that line's drawn. So that's basically all I wanted to say, but I just wanted to stress the fact that not everybody takes a standard linear journey in their career. And it's really important periodically to reflect, to consider whether you're happy, and if not, maybe sometimes making a bold decision and making a change so that you get to where you want to be. I've got some big thank yous. First of all, of course, to Nick Fox, who took a chance on a wild card and taught me the skills that meant that I could then go on and set up the imaging team in Sarah's lab. This is the Dementia Research Centre. I think it's my leaving do, and they seem to be, I don't know, singing from a hymn sheet or something. I have no idea. We've got a very youthful Ed Wild there. And I'd really like to thank all my colleagues and friends, really, in the Dementia Research Centre who supported me in the work there. This is the HD Centre. So every year we have a symposium where we get together, we get people from the lab, we have people who work on animal models, we get the clinical team, everyone comes into the same room and exchanges ideas, and I have to say it's really, really inspirational, and really just to be in the room where it happens is an incredible privilege. So thank you to all my colleagues at the HD Centre. Particular mention goes to the HD Yaz team. These are the people I work with day after day, and I love coming into the office because of this team, and I really value working with you. Of course, then, final huge thank you to Sarah Tabrizi, now CBE. Let's take a moment. We are just so proud of you, and it is a privilege, and thank you for giving me the opportunity of working in the group. Thank you to my family. These are my parents on their golden wedding anniversary. To my brother and sister and their partners. Thank you to my children, Patrick and Hope, and also Django and Oscar, who play a big part in my life. And of course, thank you to Aidan, who has supported me all the way, been my greatest champion, and supported me through many different learning experiences along the way. And finally, I really need to say something about the HD community. So on the left, we have, this is a photo taken on Saturday when we did Battersea Park Run. This was a run organised by Michaela, where we had both HD families, researchers, people from the wider institute, Selina, John and Helen, all came along, and everyone ran together to raise awareness and also raise funds for the HD Youth Organisation. So this is going to fund a scholarship so that somebody from an HD family can attend a conference, and they really benefit so much from this network of support. There's a really unique relationship, I think, between the HD community and the scientists. There's a lot of mutual respect, and you know, the HD families really are exceptional. They go through so much, and they come in year after year for assessments. They make us laugh, they make us cry, and they are our inspiration. So what's for the future? Really, what we are hoping is that we can deliver effective treatments, and what we really want to do is have the first world prevention trial in Huntington's disease. That's what we're working towards. So thank you very much for listening, and that's everything.
So, thank you, Rachel. That was a wonderful lecture, and was funny, and everything that an inaugural lecture should be. So I just want to have a vote of thanks, and for me it's an enormous honour and a real pleasure to offer the vote of thanks to Rachel. I want to say thank you for sharing your work, your insights and your journey. I think the lecture really reminded us of the impact that science can have, but also of the persistence and curiosity that can drive, and teamwork that can drive meaningful discoveries. And I think today marks a significant milestone for Rachael, and one I think that's especially fitting to celebrate, because your path to professorship has been anything but conventional, as you showed, and your career is a striking example that there is no single route into academia. And I think for many students and early career researchers will take real encouragement from your story, because it tells us, and particularly for those who may wonder whether they've taken the right path. And Rachel's illustration of the non-linear path was brilliant. And I think that journey's led you to become a leader in the field. As the head of imaging at the HD Centre, you've made an extraordinary contribution to advancing our understanding of Huntington's disease. Your works help deepen scientific knowledge, strengthen international collaborations, and move us closer to improving the lives of people and families affected by Huntington's disease. And what I think also came across so clearly was that your impact extends well beyond your scientific achievements. They're also reflected in the enormous respect and care you've earned from your colleagues, the support you provide to those you mentor, and the collaborative spirit that really underpins your work. You're a true delight to work with. I love coming into work to see you. The support you've provided over the years to colleagues and trainees has been every bit as important as the research itself, and you've mentored so many young ECRs. And an inaugural professorial lecture is a celebration of what's been achieved and also a glimpse of what's to come. And we're really fortunate that we have you working on such an important programme of research. I'd also like to thank everyone who's helped Rachel's journey, and particularly Aidan. So thank you, Aidan, for bringing Rachael back and bringing her to the HD Centre. And to all of those, the colleagues, family, friends, and all those who've supported Rachael through her career. And I think Rob said this as well, but a professorship recognises individual achievements, but that also is reflected in the many achievements that Rachael has helped with colleagues, collaborators, friends, and family members who've supported that journey. So it's very much about a group achievement, and it's really lovely to see so many of you here today. So. I'd like to say, finally, congratulations on this really well-deserved appointment. It was an outstanding lecture, and the remarkable contributions you've made to brain imaging, Huntington's disease, Alzheimer's disease, and those of us who've had the privilege of working with you know how much dedication, thoughtfulness, and determination you bring to everything you do. And this professorship is a real fitting recognition of that, and I'm truly overjoyed. So thank you. It was lovely.
Great. Colleagues, friends, thank you so much once again for joining us this evening. That brings the inaugural lectures to a close, but you're all invited to celebrate with us and have a drink outside. If I could ask the speakers and the vote of thankers to just stay behind for some photos, that would be amazing. But otherwise, just to thank both of our wonderful professors once again.